All right, hi there, Dr. P here, and in this video I'm going to talk about writing lab reports, and specifically this is for Chem 12A and 12B at Chabot College. So I know some of you viewers out there are not Chabot students. I know for sure that I have subscribers who are not from Chabot and other people who watch my videos who are not from Chabot. Thanks for tuning in. This might not be as helpful for you as it is for my students who are currently in Chem 12A or in the spring in Chem 12B. All right, so if you're going to write a lab report for Chem 12A, this is what you need to know. You'll want the following sections. You'll need an abstract. Now, what goes into the abstract? An abstract is what you did, how you did it, and summarize your results. So we will say here what you did, how you did it, and summarize results. Don't forget to include chemical reactions with structures shown. If you identified an unknown, go ahead and draw a structure of the unknown. There are some drawing programs that are available. One in particular is from ACD Labs called, Chem Sketch. Sorry about that. And that's what I use, so it's only available for Windows. There are other programs out there that you can use, or leave a blank space in your report and then draw in things by hand afterwards. Back before I started getting good with using ChemSketch, I used to, say when I was writing exams, leave spaces and then draw in the structures by hand. So it's perfectly fine to leave a space in your report, and then when you print it out, draw in the structures, write the chemical reaction. All right. Now, this stuff here should be in past tense. I know you've gotten used to doing abstracts in future tense, saying I will be, in this lab, I will be synthesizing aspirin. Instead, I want you to write them like you actually would for a journal article, in which case you'd use past tense, and you would say, in this experiment, I synthesized aspirin by whatever. Okay, so what you did, how you did it. So a brief description of like the main reactions. You don't want a lot of procedural detail. So let me write that here. Don't include a lot of procedure detail in abstract. That's what the procedure section is for. Okay. And then, you know, also how you did it, how you analyze the results. And then, you know, you'll summarize those results as well. All right, next section is going to be procedure. Excuse me. And procedure is what you did in some detail. And this could be an outline or bullet points. I'm going to go over the sample lab report that I wrote up. I made up some data for the only time in my life, and wrote up a report on the synthesis of acetaminophen. So not the synthesis of aspirin, but the synthesis of another pain reliever, acetaminophen, Tylenol. And I'll go ahead and I'll point out the different parts of the sections that I'm talking about now when I go over that. All right. You will also need data. You will want to make a data table. Or tables. And I am underlining table because yes, it should be a table. All right, you don't just want paragraph form, you want an actual table. I want to see those lines of the box-like structure. You need to include qualitative data as well. And then leave some space to include your quantitative results. So that's things like percent yield, melting point percent error, etc. And then, of course, if you have any spectra, include those. All right, and so I know you took an IR for your synthesis of aspirin, so you can include that, or you should include that. The next section, and obviously these are kind of similar to the sections that we've seen in the lab notebook. The next section is calculations. And you need to show all your work. You can do these by hand if you want to. And you could either write out your calculations by hand on another piece of paper and scan that, or just, again, leave a space in your lab report where you will add in your calculations. So you'll do the calculations in your lab notebook initially to make sure you're doing them right, and then copy them into the lab report. All right. Now, the last part is, let me get that a little, there we go. The last section that you'll have is the conclusion/discussion. And in this section, you'll want to restate the results. And discuss the results. In particular, try to explain any unexpected results. So if things didn't go quite right, or you didn't get what you were hoping to, offer some explanations. And then discuss possible sources of error. So, why did you get a 5% error on the melting point? Why was your percent yield only 10%? Okay, and then if you have any other qualitative comments about, difficulties you ran into in the lab, you can mention those as well. I know Ms. Gibson actually has a nice format, like where for a typical synthesis lab you might have 3 or 4 paragraphs. In each paragraph you're doing a different thing. So in the first paragraph you're stating your results. Second paragraph you're discussing your yield and your purity. In the third paragraph you can talk about error and ways to possibly improve the lab. And I can get you guys more information on that later. You may have a references section. This is optional. And this is any references other than lab handout. So, I've been giving you guys individual lab handouts for the procedure for each lab instead of having the commercial lab manual, instead of having a commercial lab manual. So, if you do look anything up online, go ahead and include a reference to that. Let's say you're trying to look up possible side effects of the aspirin synthesis reaction. You Google that, you find a few references, include those at the end. Okay, so now let's take a look at my synthesis of acetaminophen lab, my sample synthesis of acetaminophen lab, and put that right there, make sure that's in frame. So here we go. So you can see my abstract. In this experiment, acetaminophen was synthesized by reaction of para-aminophenol with acetic anhydride, as shown in the following reaction. So that bit there that I just highlighted, that is what you did. And then you would say by how you did it, so by reaction of para-aminophenol with acetic anhydride. Of course, we have a chemical reaction with structures. And then we have some other stuff here. I'm going to highlight these here. So a couple more sentences about how you did it. There's an additional sentence here talking about the purification. So for aspirin, you don't actually have a purification, you just crystallize it. You're not doing a recrystallization. In this lab, there was a decolorization and a crystallization, purification by recrystallization. You have, where did my Oh, there are my highlighters. Let me get my pink highlighter here. We have here percent yield was 70%, 89% recovery, and then melting point was that, and then melting point percent error was 0.5%, and then a little summary of the IR spectra here. So these are results. Okay, I mentioned the percent yield and recovery. You're just going to have a percent yield for your aspirin lab. Melting point, range, and percent error. And then a brief description of interesting peaks in the IR spectrum. All right. Next we have the procedure. And I've broken down into the procedure into different steps here. And I've done bullet points. So for example, for the procedure, I have reaction mixture, what went into the reaction mixture, heating, how it was heated, in this case in a sand bath with stirring, and what the temperature was, how things were isolated, the steps here, and just some notes here that crystallization occurred without scratching, it was allowed to complete in an ice bath, etc. And it continues, more procedure here, and then there's the decolorization procedure, crystallization, purification, or recrystallization, I should say, procedure, and then just to mention that yield and was calculated. I mentioned that the melting points were obtained and IR spectra were obtained. I mentioned what the IR spectrometer was. You can do that. We actually are using a different one. This is our old one that we're not using. We're currently using the Thermo Fisher Summit Pro. With so IR spectrometer, and I forget the name of the attachment. It's, I think, an ATR attachment. I forget exactly what it was called, but I can get you that information, or you can ask me about it. It's not important. I mean, it's kind of important, but it's not critical that you mention how the IR spectra were acquired. It's not a bad idea. It gets you in the habit of doing it for potentially other types of reports you may have to write in other classes or your future career. But yeah, don't stress about it. All right. Now we have some data. Yay, data. And so masses, volumes, and yields, melting points, and color. So I broke up the data table into two tables, one which has masses, volumes, and yields, and one melting points and colors. If you can have the data tables on the same page, that's great. Makes it easy for me to grade, but that's okay. I mentioned that IR spectra are attached, or an IR spectrum is attached, and a spectrum of the starting material is also attached for reference. I provided you with that IR. I took the IR of the pure salicylic acid and suggested that, or recommended that you take a picture, so you could include that in your report. And then calculations. I've got my full calculations here. So my percent yield, you can see I'm calculating the limiting reagent. I calculate my moles of para-aminophenol, my moles of acetic anhydride. I have one-to-one stoichiometry, so in this case the fewer moles is the para-aminophenol. So then I can calculate my percent yield. It's just a stoichiometry calculation. I know it's been a while since Chem 1A, but And then my calculated percent yield is just my actual yield divided by my theoretical yield. And I've got my calculations of the melting point. You will only have the one for the pure, okay? In this case, we're not getting a crude product, so you won't have that. Now in my discussion section here, discussion and conclusion, again, I state the results, and I'll sort of highlight results here. So yield of 70%, recovery was 89%. So I said what I did. I synthesized the acetaminophen, I got a certain yield, I got a certain percent recovery. And then I qualitatively describe or subjectively describe those results. So I'm saying here these values indicate a fairly efficient reaction in purification. What's fairly efficient? Well, if you get above 50%, that's for this class, that's not too bad. If you're like 20%, 10%, yeah, that's not very efficient. And I go on and talk about some of these here. I mentioned that unlike in the synthesis of aspirin, you can use a ferric chloride test to detect unreacted starting material. There's no simple chemical test to detect the starting material in the synthesis of acetaminophen. So that's one difference. So you wouldn't obviously mention something like that or mention this for aspirin because you have the ferric chloride test. And I mentioned some sources of loss, and it's fine to say that you lose a little bit here and there. If you know you lost something at a particular point, I knocked my reaction vial over and half of it spilled. Yeah, that goes in your discussion conclusions. I'll probably write "oops" on it, but as long as you mention it and it's a reason why you got a 10% yield, then I'm not going to take off for that. All right, let's see. And then I talk about purity here, and I mention, so in this paragraph I'm talking about purity, and so I have my IR spectrum, and then sharp peak there, although it's present in both, so I did mention that. And then I mentioned I compared the melting point and to literature value, and got a melting point percent error of 0.5%, which is pretty good. Okay, and then I also mentioned the range. So remember what things mean. A melting point range of less than 2 degrees means you've dried it pretty well, gotten rid of most of the solvent. If your melting point range is greater than 2 degrees, you have solvent present still. And if your melting point percent error is less than 2%, it's pretty pure. If your percent error is greater than 2%, then you definitely have impurities present. And then lastly here I have kind of a summary with suggestions for future labs. So, I mentioned here that, procedure was straightforward. For future labs, additional methods to test the presence of starting material, NMR, chemical tests for amines, could be added. Could do that. And actually, I realized that in the IR, you actually end up losing the NH peaks from the para-aminophenol. We'll take a look at the IR in just a sec here. Here's my reference there, is actually just the old lab manual. And then I have included my IR spectra. This is the acetaminophen, so I'm pretending that I got this spectrum. This is actually downloaded from a database. And so there's my peak for my carbonyl. And then, let me point with this here. There's my peak for my amide acetyl bondo. That peak there is from that OH. And notice that there's no peak at 16, no big peak at 1667 for the carbonyl, because there isn't one. There is a peak, I think, there for the NH2. I think it's actually there and maybe there. I'm not sure. There should be two peaks because it's a primary amine. And those are not present in the other one. So I could have mentioned that, perhaps I should have. You definitely want to make sure that you talk about your spectra in enough detail. And if you have questions about what you need to talk about as far as your spectra are concerned, ask me. Come by office hours and we can talk about it. If you got a weird result and you're having trouble explaining it, come by office hours. We'll talk about it. I can hopefully point you in the direction of coming up with some possible explanations. All right, so that was some notes on writing lab reports. I'll scan these in and post them. And yeah, good luck with your lab reports. And until next time, stay safe out there. Bye now.